Ahora sí.
Okay, so we're ready then. So it's a pleasure for me to introduce Mariana Sasitska, originally from Ukraine. Mariana is doing a postdoctoral training now at Columbia at the Zuckerman Institute for Mind, Brain, and Behavior. And originally, she studied her undergraduate studies in basic biology, general biology at Taras Shevchenko University in Kiev and in Ukraine, followed by a master and a PhD, first in genetics and second in biophysics. And then she moved to Lausanne, where she did a second PhD in molecular biology, followed by a transition to the United States, working at Columbia. You probably know that the Zuckerman Institute was organized by Axel after he got the Nobel Prize, and they have a very powerful group working on olfaction. So she has been interested in analyzing chromatin interactions in Alzheimer's, and she was working there with her mentor Lombarda, where COVID started, and they decided to take over the task of analyzing how SARS-CoV virus interacts with the genome to produce anosmia. And they produced such an interesting piece of work that she's going to explain today, that she was appointed for the Gruber Prize of Neuroscience for young fellows by the Society for Neuroscience last year. So the work has had a substantial contribution, and I'm very pleased to have Mariana today with us.
Thank you so much, Francisco, for such a generous introduction. I'm really pleased to be here and share our work with you. And just let me know if you can see my screen and if everything is working. All good?
Yes.
Okay, so let me start from there. Today I'm gonna talk about the role of chromatin architecture and its potential cause disruption as a cause of COVID-19 induced anosmia. The cover is a reference to Salvador Dali's masterpiece, which is called Sleep, with a distorted nose dominating canvas. I have chosen it to highlight a surrealistic contortion of smell perception among those losing their sense of smell. Oops, can I? Oh, okay. So why smell is important for us? We know that it's very important for most of animals, as they are using this sense for communication or hiding from predators. And apparently this mouse is not doing a great job in here. But for humans, it's also a very crucial sense, as it helps us to recognize environmental hazards such as spoiled food or gas leaking. Smell loss was associated with depression, so basically highlighting that it's an indication of general human well-being. And anosmia or smell loss, it serves as a prodrome symptom for neurodegenerative diseases such as Alzheimer's and Parkinson's. And as Francisco mentioned, I was meant to study the role of smell as a prodrome symptom for neurodegenerative disease when I joined the Lombarda's lab, when I found myself in the middle of COVID pandemic. And while all institutes were pausing their research and ramping down, there was an interesting paper published in Washington Post where the author was connecting a decrease in scented candles on Amazon when COVID hit New York City. So that was very interesting for me as a person interested in olfaction, and we decided to design a project trying to understand what's happening to those noses. And now, as we are after different waves of COVID and different strains passed by, and now we have very recent new Omicron. I want to share with you some numbers. So 103 million cases of COVID have been registered as of yesterday worldwide. And 17 to 82% are losing their sense of smell out of this 103 million. And this is quite a wide range. And I have to emphasize here that the first original Washington of Wuhan strain of SARS-CoV-2 was causing a very high level of smell loss among people infected with it, while recent Omicron is characterized with less prevalent smell loss. And then almost 6% of these people who are losing their sense of smell will struggle from permanent olfactory dysfunction. And if we will convert this percentage into numbers, it gives us up to 21 million worldwide of people struggling with smell loss. So I hope I convince you that it's quite a lot, and it's worth understanding what's going on. So as outline of my talk today, I'm going to give you a brief background on olfactory system, and mostly focusing on gene organization. As Francisco mentioned, my primarily background is more molecular genetics and biophysics. So I'm not very good neurologist here, so please apologize my ignorance in neurology. Next, we will switch to olfaction phenotypes we are observing with SARS-CoV-2 infection, and how does it impact transcriptional readouts in different cells of olfactory tissue. Then I will switch to the role of nuclear architecture and its disruption in anosmia. And lastly, if we will have time, I'm going to share some future directions and what I'm up to after this project been published. So olfactory system. Back in 1990s, Nobel Prize laureates Linda Buck and Richard Axel characterized the olfactory receptor genes. And they found that there are 1,400 olfactory receptor genes, which are organized in 69 genomic clusters in mice. For humans, we have roughly 1,000 olfactory receptor genes, which are organized into 82 genomic clusters. And basically, these numbers give us like estimate of 5% of mammalian genome is dedicated to odor recognition, meaning that it's a lot. Those genes are spread almost across all chromosomes in mammalian genome. And what is very interesting is that the expression of these genes is obeying one receptor per neuron rule, where every neuron is going to express only one specific odorant receptor. For example, can you see my pointer? Can someone tell?
Yes, yes, yes.
Thank you. So, for example, this green cell in here, let's say it's sensing banana odor. So all the cells expressing this particular odorant receptor will converge in a structure which is called glomerulus in olfactory bulb and sense banana, while all the others can be dedicated to any other type of odorants. The efforts of Lombardo's lab and others led to mapping of intergenic enhancers and olfactory receptor gene promoters, trying to understand how this one receptor per cell rule is obeyed at a molecular level. And surprisingly, the same transcriptional factors are coming there for transcriptional activation. And we figured out that all these regulatory elements and olfactory receptor gene promoters, together with shared exactly the same transcriptionally receptors, they are highly similar. And we were puzzled with the question, so how this can be solved? And what I'm explaining here is quite a bit of a challenge, where you have 69 gene clusters across 18 chromosomes, sharing 63 identical enhancers and 1,400 similar promoters, and only one allele is gonna be expressed. How that is possible? And the simplest explanation would be you need spatially segregate olfactory receptor genes and form like one cluster, which gonna be heterochromatic, where only one receptors will be introduced in euchromatic enhancer hub and got locked and expressed for entire cell life. So this idea didn't come out of a blue. It came from basics of chromatin organization. As you know, the very first or primarily basic level DNA is wrapped around histone octamers and forms 10 nanometers fiber. Then these fibers folds further and forms 30 nanometers fiber, 200 nanometers fiber, which then is organizing these topologically associated domains or TADs. The TADs are also folding further, and they are forming compartments of different potencies. They can be euchromatic or A compartment, as indicated in pink in here, or heterochromatic B compartment. That reminds something I just explained on an example of olfactory receptor genes and their enhancers. And altogether, these compartments then converge into chromosome territory, which takes decent place inside the nucleus. To probe the model we decided to apply Hi-C or genome-wide chromosome conformational capture technique. I will briefly describe you what that is. So basically, we are gluing together chromatin with all other proteins inside the nucleus. Then this cross-linked chromatin is cut with restriction enzymes, holding two parts of genome which are far apart in linear genome, but close in a 3D volume of nuclei together. Then overhangs are filled with biotinylated mark, ligated, and after shearing, we can pull these hybrid reads where we have one piece coming from one loci and the other one is coming from completely different loci. And the analysis is revealing something which I really like. You can think of it as of Google Map. That's the analogy is coming from Robinson, who is working at the Baylor College of Medicine. That's why you can see Texas State in here. So these rectangles across the diagonal, these are separate chromosomes. And you can think of it as of entire globe view. Then we can zoom in. And when we are zooming in, we can see that the chromosome, basically, its organization is a plate where you can see red regions which are enriched in interactions. Then the gradient indicates the strength of interaction. And if some regions are avoiding one another, we have white, no interaction. So that is some kind of like a state view. But if we want to go deeper, we can go to the level of topologically associated domains, which basically is like hundreds of kilobases up to the megabase, which would give us like you can think of it as of the region. And finally, we can zoom in so greatly that we can see particular loci or loops or gene, how it's getting expressed. And that is Baylor College of Medicine in this analogy. Okay, so we performed genome-wide chromosome conformational capture on mature olfactory sensory neurons. And here what we see. So this is how real high-C maps looking like, with these squares across a diagonal depicting interaction in between topologically associated domains with enrichment and depletion in between contacts of different loci. So that's what we did for olfactory sensory neurons. And here I'm showing this triangle. It's basically the same as this triangle. Since the matrix is symmetrical, I'm just showing one part of it just for better representation. And red squares underneath is the genomic location of olfactory receptor gene clusters. And here you can appreciate that two clusters located on chromosome 2 are getting engaged in extensive interaction, showing up in here with this dark red spot. But also we notice that there are intensive and highly robust interaction, not only within olfactory receptor gene clusters within the same chromosome, but also between different chromosomes. So these arrows are indicating olfactory receptor gene clusters located on chromosome 9 and chromosome 2 respectively. And when we will zoom in, we can see that there is a hotspot of interactions, and this broader regions of interaction. So the broader regions are olfactory receptor genes, while this one is intergenic enhancer euchromatic hub, meaning that the model I was describing at the very beginning is actually proved by this high-C experiment. Now, I want to give you another example of experiment which was done in the lab in the past. So basically here what you see is normal olfactory sensory receptor cell. And usually they are not expressing LBR or lamin B receptor. And that results in inverted nuclei morphology, where the heterochromatin is centered within the nuclei. This is this bright orange spot, while euchromatin is pushed towards the periphery. And when we are doing high-C on these cells, and then subsetting only interaction of our cluster genes, this is what we see. Every olfactory receptor gene cluster is color-coded here at the bars on top and left. And the brighter the dot in here, the stronger the interaction between clusters are. And again, you can appreciate that olfactory receptor genes which are belonging to the same cluster tend to interact stronger. But also there is this robust interaction in between different chromosomes. So next, what was done, the LBR receptor, which is normally is not expressed in mature OSNs, was ectopically expressed. And what do we see here? We see that the fried egg morphology of olfactory sensory neuron is lost. And together with that, the context in between olfactory receptor gene clusters is going dissipated. But also we were wondering if that does have an effect on transcriptional readout. And here is RNA-seq analysis of olfactory receptor gene expression in LBR positive OSNs versus control OSNs which do not express LBR. And you can appreciate here that OR expression is dramatically downregulated. I hope that I have convinced you with that, that genome architecture is crucial for transcriptional readout and production of olfactory receptor genes, which are crucial for olfaction as general. But now let's get back to SARS-CoV-2 and olfactory neurons and olfactory epithelium. So when pandemic has started, we all were puzzled with the fact that there is olfactory dysfunction in COVID-positive patients. But how is it possible if olfactory sensory neurons are not expressing AC2 receptor? AC2 receptor is one of receptors which is the entry gate of virus into the organism. So here I will briefly explain you how olfactory epithelium is organized. The very basic cell layer here marked in brown is horizontal basal cells, or HBCs. They do express AC2 receptor. Then we have a layer of globosal cells, which are able to cell renew and give rise to substanticular cells and other OSN progenitors indicated here in green. Then in pink, it's mature olfactory sensory neurons, which are covered with substanticular protective cell layer. And these cells, again, are expressing AC2 receptor. So we decided to take hamster as our experimental model, as hamsters are having highly similar to humans AC2 receptor and getting infected with COVID, while mice are really resistant to SARS-CoV-2 infection. And the first things we were interested in, if there was a tropism of SARS-CoV-2 to olfactory epithelium. And this was done in collaboration with amazing former PhD student in a lab and now a postdoctoral research fellow at Rockefeller University, Albana Kodra, who did a series of immunofluorescence, which was combined with RNA in situ hybridization to figure out which type of cells is getting infected. And here on the panels in green, I'm showing OMP, or olfactory major protein, which is a marker for mature olfactory sensory neurons. In magenta, these are SARS-CoV-2. And here is overlaid together with DAPI, which is marking just nuclei of the cells. You can appreciate that there is lack of collocalization of the two signals. And here is a quantification and correlation in between green and magenta channels is rather negative than positive. So that gave us a hint that it's not really likely that olfactory sensory neurons are getting infected with SARS-CoV-2. But imaging was not enough to be convinced that olfactory sensory neurons are not getting infected. And at the time, there was a paper about another neuropilin receptor, which might be an entry gate for neuronal epithelium. So we decided to apply another orthogonal approach, which is a single-cell RNA-seq analysis to figure out which cell populations are getting infected with SARS-CoV-2. And I like to show this image because I'm thinking of a single-cell RNA-seq as of unblending smoothie. You are starting with cell mixture, and after sequencing it, you are able to distinguish which cell it was coming from. And here I'm just comparing, showing you a comparison between two approaches, which we took in our studies. The first one is single-cell RNA-seq analysis, which enables to characterize each cell type which is present in tissue originally, and then with dimensionality reduction UMA plots, we can position different cell populations and figure out what they are. While bulk RNA-seq is meant to take RNA from entire this tissue without splitting into separate cells, and it gives us an average gene expression from all cells, where heterogeneity of cell population is masked, but it's very useful to apply this method when you are talking about genes which are lowly expressed like olfactory receptors, and they are all unique. So that was the way to go with figuring out what's happening with our expression. So now, does COVID have a tropism to OSNs? We did a time course of SARS-CoV-2 infection, starting with smoke with 0 virus, and then 1 day post-infection, 3 days post-infection, and 10 days post-infection. And we did single-cell RNA-seq analysis. And here you can appreciate the distribution of cells across all 4 conditions is similar. We can detect a lot of olfactory sensory neurons, their precursors, and other cell types which are present in olfactory epithelium. But what is noticeable here is that some cell population are different in 3 conditions. For example, we have a lot of substanticular or supporting cells in mock hamster. Then amount of these cells is decreasing 1 day post-infection. And here you can appreciate depletion of these cells 3 days post-infection when we observe the maximal viral load. And then restoration 10-day post-infection when infection is already cleared. And together with that, we observe a massive infiltration of microglia and immunity cells like T cells, B cells, and macrophages when viral load is the highest in infected animals. So now that all is good, but where is the virus? Here I'm showing on this human plot with red counts all SARS-CoV-2 and transcript we were able to detect. And I have to mention that N transcript is one of the highest expressed in among other SARS-CoV-2 transcripts. And what we observe that virus is present in these immunity cells, microglia, TB, and macrophages, but also in substanticular and HBC cells, which are 2 cell types of olfactory epithelium which express AC2 receptor. Then we looked at distribution of SARS-CoV-2 and transcript across different cell types and figure out that only tiny part, which is not exceeding 5% of cells of olfactory neurons is having virus. So it's such a tiny amount which cannot account for the fact that of anosmia. So then we decided to look more carefully into cell autonomous changes which are happening in substanticular cells as they are getting infected with SARS-CoV-2, and non-cell autonomous changes which are happening in OSNs as they are not getting infected. And here with these cartoons, I'm highlighting which cell types we are looking at, and I will continue doing that along the presentation. So, as expected, in substanticular cells, we are observing massive upregulation of different COVID-2 transcript, but together with it, we are observing upregulation of interferon gamma pathway responsible genes, and downregulation of markers of substanticular cells, which is not a surprise as we know that they are getting infected and probably dying. As we saw that there is a decrease in cell number. But what happens with OSNs? While they are not getting infected, we observe this massive upregulation of inflammatory responsible genes like AF2AK2, which is sensing double-stranded RNA and acting via toll-like receptors. The same ISG15, which is highly upregulated in substanticular cells. And again, I'm emphasizing that these cells are not getting infected, but they are reacting, their immunity response is similar to the cells which are getting infected. Next, we were wondering what's happening with olfactory signal transduction. And here I'm showing you the expression of critical signal transduction genes, which are helping to pass the signal from olfactory receptor on the cell surface up to olfactory bulb. And again, we are looking at them at mature olfactory sensory neurons. And you can appreciate that all of them are getting downregulated as infection progresses. And the most dramatic effect we are observing at day 3 post-infection when viral load is peaking. And together with that, what we observe again in mature olfactory sensory neurons is that there is massive upregulation of inflammatory and immunity response genes of interferon gamma pathway in this case, like IRF7, IRF9, IF2AK2, MX1. They are all getting upregulated, and that happens when the viral load is peaking. Now, as I already mentioned, we cannot measure everything at a single cell level, especially with the chemistry used for single cell, which is 5' and good for capturing viral counts. It's not good to capture 3' UTRs, which are greatly annotated for olfactory receptor genes. So we were not able to address the question what's happening with ORs at single cell level. So we did bulk RNA-seq. And here you can observe how local change of OR expression gene is changing when viral infection is progressing. And 4 days post-infection, the downregulation is the most dramatic. And that is coinciding with downregulation of roughly 50 OSN marker genes. So it's not only olfactory receptor genes which are stopping their production, but also other genes which are involved in olfaction as a process are getting downregulated. And here I'm summarizing what's happening with olfactory transduction, which is getting suppressed very dramatically day 4 post-infection, and that coincides with massive upregulation of immunity or interferon pathway in infected hamsters. So that was our model organism hamster, and we were wondering what happened in humans. And here I have to emphasize a seminal effort of ENT here at New York Presbyterian, Jonathan Over the West, who volunteered to collect olfactory epithelium from people who passed away from COVID and volunteered to donate their specimens for science. That was very tricky at the time because we didn't know, it happened just at the beginning of pandemic, and we didn't know how infectious these samples were. And you need to go through cranial bones to get into olfactory epithelium in humans. And I have to say that it's completely different from what it looks in rodent species, where olfactory epithelium is looking like a decent structure, and you can easily distinguish it from respiratory epithelium. In humans, there is no separation between, it's just gradually a respiratory epithelium is going into olfactory epithelium. And that was very important at the time, as respiratory epithelium is enriched in SC2 receptor. And that led to different publications coming and saying that the olfactory epithelium is getting infected, while that was just a confusion between the two. So we decided firstly to characterize properly olfactory epithelium. And for that, again, we took 2 different orthogonal approaches. Immunohistochemistry, where here LDB1 and OMP are markers for olfactory sensory neurons, and DAPI stains suggest nuclei. So to confirm that the region we are looking is indeed enriched in olfactory neurons. The other approach we took was single-cell RNA-seq analysis, which again revealed that we have decent population of olfactory neurons in our specimen. And then again, Albana did RNA-ish, trying to understand if in humans we see the signal collocalizing, signal from collocalizing with olfactory sensory neurons, which are marked here in green and COVID in magenta. You can appreciate a very apical localization of COVID transcripts. And again, there is no collocalization of green and magenta together, meaning that like hamster, human epithelium is not getting infected with SARS-CoV-2. Next, we decided to measure our genes expression across the samples. Total, we had 23 samples, 18 of which were COVID positive and 5 controls. And what we observe, the control samples are marked here in blue, while reds are showing COVID samples, and you can appreciate that there is dramatic reduction in olfactory receptor gene counts in infected samples. And that's not due to the lack of cells or not correct region, as olfactory sensory neuron genes stay the same across all the samples, and the same is true for olfactory epithelium general markers, which also are looking similar across all the samples. There was only one sample, a control sample, which took our attention as it had very low OR counts. Here it's marked in this light blue, and we decided to take a closer look what's going on in that sample. And it happens that the person was infected with non-SARS coronavirus, which was known to cause smell loss and responsible for epidemic in Europe, which was in 1899, if I'm not mistaken. That was interesting fact, but this sample was taken out of our cohort for further analysis. And again, we checked what's happening with olfactory signaling genes and noticed that there is a dramatic downregulation of all components of signaling pathway in samples infected with COVID. And some R expression is also dramatically downregulated in COVID positive samples. Now, I want to draw your attention back into chromatin organization. I am emphasized that chromatin is compartmentalized inside the nucleus. It has different levels of its organization, from loop organization to contact domains, and then compartments, the chromosomal territories, and that's how we can approach it and measure it. We are doing high C, and that's actually what we have done for human and hamster specimen. So here I'm showing you again a time course for hamster, where we are taking a MOCO control sample, 1 day, 3 days, and 10 days post-infection. Green bars on the top are depicting genomic localization of olfactory genes cluster in hamster. And now we are looking on whole chromosome view. In this case, it's chromosome 16. And in arrow, I'm pointing your attention to the cluster genes, which is located in here. And you can see that this how many is the 2 and 5 clusters in here interact robustly in a control sample. And then when we are going along the infection progression, you can see how that dissipates when amount of virus is peaking. And that is also true not only for intrachromosome context, but also interchromosome context. Here, I'm showing you exactly the same, but now interaction between chromosome 16 and chromosome 17, and you can see this numbered or cluster genes. And again, their interactions are getting dissipated when COVID infection reaches its maximal level. And they start kind of slightly restored then 10 post-infection. And I have to emphasize that day 10 post-infection is the time point when virus is absolutely cleared from infected animals. Then what I did, I subtracted all olfactory receptor genes cluster and I visualized it as a heat map. And here you can see that effect, which I was showing in the previous slide, even more pronouncedly. You see this robust interaction within the same chromosome and intensive interaction in between different chromosomes, and they are getting disrupted by day 3 post-infection and again getting restored. Day 10 post-infection, but not completely, still not reaching the same level as in control animal. And here's a quantification with significance scores. Next, what we did, we checked if that's the case for human samples, and here it's 2 control samples compared to 4 COVID-infected samples. And when you look at these squares in here, you can appreciate that in COVID-infected samples, the interactions are much weaker, and the confirmation of that with quantification. But that is also true not only for particular olfactory receptor gene clusters, but also for topologically associated domains. So we apply AI for calling topological domains on these maps. And here the blue lines are showing topologically associated domains called in control samples, while on the other side of the diagonal, I'm showing you now SARS-CoV-2 infected samples. And as you can see, this unsupervised approach was able to depict much less contact domains compared in infected samples compared to control ones. And here's another representation of dissipation of compartments between different genes. And in this case, we are talking about compartments which are formed between genes involved in olfactory signal transduction, which are getting dissipated. So now we know that olfactory receptor genes are getting downregulated, signaling component genes are getting downregulated. The potential cause for that is disorganization of underlying chromatin topology. Now we were puzzling, is there a circulating signal that induces these processes? And to answer that question, we did this elegant experiment where heavily infected hamster with SARS-CoV-2 was used to collect serum, which then was irradiated with ultraviolet to be sure that all virus is deactivated. And then we applied the serum to naive hamster, and 12.5 hours later, we performed Hi-C. Before doing that, we did RNA-seq on this serum just to justify that there is no SARS-CoV-2 counts. And here I'm showing you comparison of the two. This is SARS-CoV-2 infected hamster serum compared to 1 day post-infection. And these are contact matrices showing interaction between olfactory receptor genes in control hamster compared to infected SARS-CoV-2 serum applied hamster. And you can see that, again, there is the same effect, that the interaction in between olfactory receptor genes is downregulated. So with that, we were able to draw this model. where olfaction as a process is relied not only on olfactory receptor proteins, which detects different odors, but also on very sophisticated yet very fragile interaction between olfactory receptor genes, which are organized in inactive OR compartment, and active enhancer hub compartment, which grants monogenic monoallelic expression of OR inside of every olfactory sensory neuron. And upon SARS-CoV-2 infection, what we observe, the first thing is that the tissue is not as integer as in normal condition as two cell types, in particular HBCs and AC2, which are and SAS, which are AC2 positive, are getting infected. And we anticipate that signals, maybe even cytokines, which are released by these infected cells, can suppress somehow or impact olfactory sensory neurons in a way that chromatin is getting disrupted, and thus it prevents a transcriptional readout for olfactory receptor gene. And eventually it leads to olfactory dysfunction in general. So that was the study we did regarding smell loss in COVID positive patients, but we didn't figure out what is that molecule triggering nuclear architecture reorganization or dissipation of contacts between olfactory receptor genes. So I'm gonna share some future directions I'm taking right now. And the first thing we decided to do is to examine people who are infected with COVID-2. Now, as most of people are vaccinated and it's not as life-threatening as it was at the beginning of pandemic, we have designed a minimal invasion a protocol for acquiring human olfactory epithelium. It's not invasive. It's basically we are placing polypropylene sponge to the roof of cribriform plate and then soaking the mucus from that area and extracting RNA. And after that, we can sequence these samples. And that reveals that basically in these samples, we have whole heterogeneity of olfactory epithelium cell populations, starting with substanticular or supporting cells and mature OSNs, progenitors, stem cells, GBCs. All cell types are present there. And moreover, we are able to measure amount of olfactory receptors which are expressed in the sample. So in these three cases of volunteers, we can see that these two are having much lower expression of olfactory receptor genes compared to this one. And we know that these two persons were struggling from olfactory dysfunction post-COVID, while this one never had such a case. So basically, this new approach is opening new routes for better understanding what's happening with human olfactory epithelium upon COVID infection. Now we can do it basically in real time and less stressful condition. And the other alternative approach we are taking is a gain of function screen for SARS-CoV-2 different ORFs in pseudotype lentiviruses. So basically what I'm doing, I'm taking either lentivirus, which is having M-cherry or whatever reporter protein in it, or it does have different components of COVID transcript. I picked ORF8 as the one which is most known for now, and it's known to be involved in inflammasome formation. And then I'm using it in different combination and applying to mock mice. And whenever they are receiving the pseudotype lentivirus, this is what I can observe. If mice are receiving spike pseudotype lentivirus, I'm observing the substanticular cells are getting downregulated, which is in line with what is observed with substanticular cells during real COVID infection. And next, when I'm using a combination of SARS-CoV-2 together with spike on lentivirus capsid, that's a very interesting thing I'm observing, that if it's spike only, then our expression is mostly not changed, but it's getting downregulated if it's combination of spike and ORF8. And this is a comparison with hamster, which is infected with real COVID. So basically that gives a hint that potentially ORF8 somehow can trigger all the effects we are observing in OSNs upon COVID infection. But again, that's a very preliminary data, which I put together just yesterday. So with that, I hope that I managed to convince you that having olfaction as a phenotype and having tools such high CO genome conformational capture, we can approach not only the causes of smell loss in infection disease, as I have shown you on an example with COVID, but potentially we can use it as a prognostic marker for a neurodegenerative disease such as Alzheimer's. So I'm still planning to work on smell loss and OSN's nuclear organization in Alzheimer patients. And with that, I would like to thank these three brave souls, Albana, Jonathan, and my PI, Stavros Lombardas, without whom this project won't ever happen. They were there for me days and nights and doing whatsoever was needed. And also, I would like to acknowledge our collaborators from Columbia Biobank, New York University, and Mount Sinai, who are helping with hamster infection and tissue processing. I would like to thank Lombardas Lab and funding and you for your attention. And with that, I'm happy to take any questions you have.
Thank you very much, Mariana. It was a lovely seminar with such relevant results. So let's see if there are any questions among the audience.
Do you want me to stop share screen or you think?
Yes, that would be nice. Thank you.
Okay.
Is there any question? Could you please raise your hands? So in the meanwhile, Mariana, this mechanism that you find, you mentioned already something, and mine is of course an ignorant question. Do you think this predicts the mechanism by which other viruses are producing similar responses in olfaction?
That's actually a very good question. And we were thinking about probing some animals which are infected, for example, with influenza A virus, which is also known to cause olfactory dysfunction. And for some people, it happens permanently. So having that in mind, potentially, yes. But I don't know how influenza A is interacting with olfactory neurons. With SARS, we know that they are not getting infected, but with influenza A, it may be the case that cells are getting infected, and it's like direct consequence, as in case with SARS, cells and COVID.
And the reason why some cells, not the others, are infected by the virus is the presence of the receptors at the plasma membrane. No other factor is taking place on that.
Yes, it's exactly because of presence of AC2, or the other one is TMPRSS2, but both any cell types except of SAS and this STEM HBC cells in olfactory epithelium are having that. So that must be something indirect, and potentially it can be replicating ORF8 RNA, because we see this coincidence that ORF8 has an effect when we are doing this pseudotype virus. And at single cell data set, we are observing that there is massive increase of AF to AK2 gene, which is basically responsible for capturing double-stranded RNA, which is getting into the cell. So maybe that is the thing, but still I'm lacking, like evidence that it's double-stranded RNA, which is harming OSN so badly.
Okay, thank you. Are there any other questions? Susana?
Yes, thank you. Thank you very much. Very interesting seminar. I was just wondering if, so have you tested also, is it recovered the pattern of expression in the people that recover the smell?
That's actually, that's fantastic question. And that's something which ongoing, because now we want to have like 3 categories of people: the ones which are having like right now COVID and smell dysfunction control group, and also those ones who are experienced long COVID or who never recovered post infection. But that's going, and like we have designed this non-invasive approach. So it's not painful. It's not even a biopsy. It's to easily get this sample. So that was the aim for that protocol to be established. So it's kind of, it's in a list, but I don't have any data to share yet.
Okay, because since people recover, I imagine that so it would be very interesting to analyze the dynamics in the nucleus to recover all the topology.
Yeah, exactly. I'm really curious to see what's happening with those nuclei, especially in patients who are still struggling with smell loss. But here's one caveat. As HBCs, which are stem cells, they do have a AC2 receptor, and they are getting infected. So there can be a case that all those cells are infected and dead. So basically, what could happen is that there is no olfactory epithelium anymore. And
yeah, that's happening in the people that does not recover. But I am more curious what is going on inside of the neurons of the people that recovers the smell. How can it be recovered? I mean,
how can it be recovered? We have done some kind of survey studies with our ENT, and what we are observing is that there are a lot of deviation of normal smell perception in people recovered from COVID. So there are conditions which are called parosmia, when people are sensing smells differently. So basically, the thing which is basically chocolate is smelling, let's say, like banana.
okay.
What I think is happening there is whenever genome architecture returned back into place, the wrong olfactory receptors got into olfactory receptor hub, but projections is still remaining to the glomerulus, which is sensing chocolate, but not banana. You see, some kind of swamp has happened in between olfactory receptor gene, which has to be expressed. The other thing, which is also getting very prevalent is hyposmia, when people are smelling, but not to the extent it was before COVID. But all this is just indicating to me that we need to develop some kind of tools for objective smell testing, because right now everything is self-reporting, which is hard to rely on, right? So I'm really curious what's happening to those neurons, but for now I don't have an answer.
Okay. Thank you very much. Congratulations.
A question that's related to Susana's is: should the recovery of the smelling capability be related to the regular rate of replacement of the olfactory neurons, or is there an additional mechanism?
That's just a fantastic question, and my answer would be I don't know. I don't know because we don't know to which extent the recovery is happening. And what's known is that while we are aging, the thickness of olfactory epithelium layer is decreasing, and that is associated with worsening of smell perception with age. But is that the similar effect which COVID does? Or it's some kind of different issue? That I have no idea.
Yeah. So how damaged are the precursor cells during COVID?
How about other cells? Do you mean if they are changing their patterns?
Yeah, because probably the precursor cells are not deeply damaged by COVID, and they just continue producing.
So that's again the caveat, because the precursors, the stem cells, which are giving rise to all the precursors, they are getting infected. And we have to keep in mind that olfactory epithelium, these neuronal cells, they have this duality between being neuronal cell and epithelial cell, and they are not with us like for forever. They are shedding, and the replacement of olfactory sensory neurons is happening every, if I'm not mistaken, 3 months. So every 3 months we have new olfactory neurons. And if the niche of stem cells is gone because they got infected and died, basically there is nothing to replace from, right? And that is really irreversible if there is no and actually I had a couple of long post-COVID samples where, like the region is right, the place is right, and there is not a single olfactory sensory neuron in that specimen.
Wow.
So with that, I think that it's possible that stem cells are dying. The others are, with the time, they are meant to be replaced, and they also are dying with the time, right? And there is nothing to reproduce new olfactory neurons, and with that you can do nothing, basically.
Okay. Well, thank you very much. I'd like to thank Roman for the organization, and moreover to Mariana for the wonderful seminar, so clearly presented and so elegantly. Thank you very much.
Yeah, I really enjoyed it, and thank you for the invitation. Thank you, Roman, and especially thank you, Francisco, for giving me this opportunity to present.
Hopefully next time we'll be in
present.
Oh, there's some. Susana is clapping, so I think you have a very deserved clapping. Thank you very much.
Thank you so much. Thank you, and have a good day.
Same to you. Same to you. Bye-bye.
Bye-bye.
Gracias, Roman.
Súper bien, pues.